welcome to all of you in the next class of 5 kingdom classification. In our previous class, we studied different aspects of the kingdom Monera and we studied bacteria in detail. Okay, in this class, we are going to study about eukaryotes. Okay, eukaryotes which are present in kingdom Protista. Okay, so kingdom Protista is the next kingdom of our 5 kingdom classification proposed by R.H. Whitaker. First one was Monera and second one is Protista. Okay, and then rest we will study in our further classes. In this class we will study about Protista. So let us start, my dear students. First of all, let's see a few basic characteristics of this kingdom. Okay, characteristic features of this kingdom. So first of all, I will like to share two images with you. Okay, this image is a protista. Okay, it is in particularly it is called paramecium, and no, it is called euglena. I'm sorry, this one is paramecium. Yeah, it is called euglena and paramecium. These are two types of unicellular organisms which come under this kingdom. Okay, let's see a few category characteristics. The characteristics are they are eukaryotic, single celled organisms. Eukaryotic means the cells, internal organ, internal boundaries of cells will be present. Okay, and the nucleus will be in a membrane, and other cell organelles will also be present. Let's see what is a eukaryotic cell. The cell has a well organized nucleus with one or two nuclei. Okay, the cells of eukaryotic cell have a very well organized nucleus with one or two nuclei. Okay, next thing is the cell possesses mitochondria. Cells of this kingdom possesses mitochondria. That means they are little bit evolved than prokaryotic cells. Okay, or prokaryotes. So they possess mitochondria. What else? They are either autotrophic or heterotrophic in nature. As you can see in this picture, we have chloroplasts. Okay, in this picture we have chloroplasts. In the left one here, chloroplasts, and here chloroplast is not present there, so it can be autotrophic. This left organism, which is euglena, will be autotrophic, and the right one will be heterotrophic because it does not have chloroplasts for doing photosynthesis. Okay, so they can be either autotrophic or heterotrophic. Okay, next is there is a special structure called contractile vacuole. Contractile vacuole is a special structure. As you can see in both these images, contractile vacuole is present. Okay, and it helps in many ways to the plants. It helps for doing excretion, for making food vacuoles, and these things vacuoles. Okay, we will study these vacuoles in details in further slides. By now just see that contractile vacuole is present in these cells. Okay, and next thing is which helps in waste removal of wastes. Okay, it is a very important feature of contractile vacuole that it helps in removal of waste. Pseudopodia, cilia or flagella are present for locomotion. My dear students, in the previous kingdom where bacteria were there, nothing was present for locomotion. Okay, but here very clearly defined pseudopodia, cilia or flagella are present for locomotion. As you can see in this picture, the organism which is on the right have small hair like projections across the body. These projections are called cilia. As you can also see here, these projections are called cilia, and this is present in paramecium. And on the left side where euglena is given, you can see a green flagellum going out of the cell, a hair like long hair like projection, flagellum. It helps for locomotion. Okay, So these cells have what is pseudopodia, we will see in a while. So these things help in locomotion also. They reproduce asexually by binary fission or multiple fission. Since they are still unicellular, they don't have different separate or specified sex organs, so they also multiply by simple binary fission or multiple fission. Okay, they either divide in two cells or in many cells depending upon the situation which we will study. Okay, some common protists are amoeba, paramecium, and euglena. These are some common protists. Okay, so paramecium and amoeba we have, paramecium and euglena we have seen. Amoeba we will see in a while. Okay, okay. Prokaryotes, here a little thing is that a microscopic single-celled organism having a distinct nucleus and cell organelles. Those microscopic single-celled organisms which have a distinct nucleus, I mean that can be which can be identified very clearly, a distinct nucleus and other cell organelles, distinct cell organelles, that type of single-celled organism are kept under this category, which is Protista. Okay, let's move further and see these organisms in detail. Euglena, we will see. Euglena looks like this, my dear students. They are small green unicellular organisms which have their own flagella, as you can see in this picture. The flagella of this euglena can be seen clearly, and how the cell is moving, and how the cells are actually. These are real images taken by electron microscope, my dear students. So you can see how these cells are, how they move around, how they function, okay, how they look like. Okay, let's see this also, paramecium. This is a paramecium taken in some fluid. Let's see one more image here. This one is also a paramecium, real image of a paramecium. It is moving with the help of cilia, which is present all across the body. Okay, okay, so let's move further. Amoeba, now let's see. We in this chapter we have to study amoeba in details. Okay, paramecium and euglena we have seen for a basic idea, and in this section, in this chapter, we have to study amoeba in complete details. So let us start with that. This is, my dear students, an amoeba. You can see how it is moving and how it looks. This is a amoeba, a true amoeba. Okay, so amoebas are also eukaryotic. They have distinct nucleus and things like that. Different cell organelles, as you can see in this picture, different cell organelles can be seen. Okay, so let us study a few things about it. Amoeba is unicellular free living animals. Okay, amoeba is unicellular free living animal. Okay, yeah, amoeba are unicellular free living animals. It lives in freshwater ponds, lakes, and ditches. It lives in freshwater. Amoeba lives in freshwater. Okay, ponds, lakes, and small ditches which are there in the rainy season. Nowadays, we can see a lot of ditches here and there whenever it rains. The cells are filled with protoplasm and does not have a distinct shape. These cells are filled with protoplasm. They are jelly like. Okay, they don't have a very fixed shape. They keep on changing their shapes very easily. Okay, okay. Pseudopodia are finger like cytoplasmic projections that helps to move around, feed, and excrete. Pseudopodia are the main things. As you can see now that in this image on the right hand side, small finger like projections are growing from the body of this amoeba. So these finger like projections are called pseudopodia. It helps them to move around, feed, and excrete. Okay, my dear students. The cells have a distinct nucleus, a contractile vacuole for excretion, and one or two food vacuoles are also there. Okay, how these food vacuoles are formed and how this contractile vacuole helps in excretion, we will see in details. Okay, so now let us continue. Let us move one step further and see a diagram of amoeba. This is a diagrammatic representation of an amoeba cell. Okay, it has pseudopodia. An amoeba uses pseudopodia to move and feed pseudopods from when pseudopods from form when cytoplasm flow towards one direction and the amoeba follows. Okay, it simply flows around. We will see here in a minute. Cell membrane. What is cell membrane? Because the cells because the cell membrane is very thin and flexible, an amoeba shape changes continuously. It does not have a cell wall like in bacteria. It has a cell membrane and its cells keep on the shape changing their shape continuously. Okay, contractile vacuole is also present, which is very distinct characteristic of this kingdom. The contractile vacuole collects excess water from the cytoplasm and expels it from the cell. All type of waste material, not only excess water but all type of waste materials, are excreted through these contractile vacuoles. Food vacuoles are also present. When the end of two pseudopods fuse, they form a food vacuole. Food is broken down inside the food vacuole in the cytoplasm. This is the cytoplasm, the liquid which is filled inside the cell. Nucleus is also there. The nucleus controls the cell's function and is involved in reproduction. Amoebas usually reproduce by binary fission if the situation is favorable. Okay. Now we will study in detail about locomotion in amoeba. Amoeba moves with the help of pseudopodia. By the time we are known, amoeba can form pseudopodia in any direction and it flows in the direction of pseudopodia. What does this statement is saying? It will be very clear after seeing this image. As you can see, this organism is forming pseudopodias in the on right hand side. Okay, and it is flowing in that direction. Okay, and other cell organelles suspended you can also see, and nucleus you can also see, contractile vacuole you can also see behind nucleus. Okay, so cells move like this, forming pseudopodia and moving forward. Okay, I hope this is very clear to you all how an amoeba moves forward. Okay, pseudopodia or false foot. Okay, nucleus and other cell organelles. Okay, okay. Now we have this sort of movement is has been given a name, and that name is amoeboid movement. If something moves like this by forming pseudopodia type of thing and flowing in one direction, it is called amoeboid movement. Okay. Okay. Next point is nutrition in amoeba. Okay, so how does it takes place? For nutrition, we will see amoeba is found in fresh water. It feeds on microscopic algae, bacteria, plant cells, and other microscopic protozoa present in water. Everything which is basically smaller than amoeba, it eats, and which is organic, which is something living or something from living organisms. It cannot eat rocks and soil. It eats organic cells which is smaller than amoeba. Okay, now see this how it happens. Okay, okay, as you can see here in this image, a small grain type of thing is there. That is the food which this amoeba is trying to engulf. Okay, and what it will do, it will go there, it will detect that yes, this is food we want to intake, and it will divide its pseudopodia in two directions, cover, simply go divide it and cover it, and then make a food vacuole and then proceed, and that food vacuole will be carried along with the cell. Okay, for digestion. So this is how it works. Amoeba intakes its food with the help of pseudopodia. Okay, I hope this is clear. How does ingestion is happening here? Let's understand different aspects of digestion in detail. When an amoeba senses a food particle, it encircles the particle by forming two pseudopodia around it. When the food particle is completely encircled, it is engulfed in the form of a vacuole called food vacuole. Okay, this is very clear. It will go, it will sense that yeah, this is food, this is tasty, it smells nice, it looks good, I'm going to eat it. And what he will, or it will do, it will make out two hands, and it will capture that food, and it will ingest it, and forming a vacuole in which the digestion will take place, and it will move along. Okay. Okay. What is digestion? How digestion takes place? So, digestive juices are secreted by the cell to break the food particle present inside the food vacuole into simple molecules. Just like every organism does digestion, they secrete some juices on the foods to break them down into simpler molecules for absorption. Same process is also done here. What is absorption? So the simple molecules formed after digestion of food is absorbed by cytoplasm. The undigested food particles remain in the food vacuole. Okay. So the useful things they will absorb just like us. We also do the same thing. After digestion, absorption is taking place in the small intestine, where we absorb all the nutrients and minerals and vitamins and all those things. Same type of function is activities amoeba are also doing. They will break the food particles, and they will absorb the useful materials, and they will leave the unuseful materials which are not useful inside the food vacuole. Okay. Now assimilation. Amoeba uses the simple molecules which are formed as a result of digestion. Amoebas uses the simple molecules. Okay. So what will it do? It will use those molecules, like suppose some protein came, it will use it for making some organelles and something like that. Energy came, it will use it. Glucose came, water came, it will use it. Whatever comes, it will use it after absorption. That process of using absorbed nutrients are called assimilation. It also happens in human beings and other animals also. Agitation is also very common process of phenomena of digestion. In this stage, the undigested portion of the food stored in the food vacuole is removed from the body by rupturing the cell membrane. Okay. Now, by rupturing the cell membrane means it will go on the vacuole will go near the boundary of the cell. Okay. And then the cell membrane will rupture, and everything is excreted out along with the vacuole's inner membrane will come and touch that rupture and make it again, the cell will become intact. Okay. Okay. Now let's see respiration, how it happens. For respiration, diffusion is used. Simply, oxygen will be absorbed from the atmosphere and carbon dioxide will be released in the atmosphere. Let's see in detail. Amoeba respiration occurs through the general body surface by the process of diffusion. Okay. Oxygen from the water body diffuse inside the food, inside the body of the animal. This oxygen is used to break down food to release energy. Okay. Oxygen is used definitely for breaking down of food to release energy, the same process which happens inside us, which is called respiration. Okay. During this process, carbon dioxide is formed, which diffuses out of the body of the animal into the surrounding water. It lives in fresh water, so this process is happening inside water. Carbon dioxide will be released and oxygen will be absorbed from the surrounding water. Okay. Excretion, my dear students. What is excretion? So excretion we will study in detail here, that this process I was talking about. See, excess of water is released, that these vacuoles, these contractile vacuole, one of them which was fully loaded came in contact, with the boundary when it was full, and it simply explodes exploded outside. And then this inner membrane, after explosion, the inner membrane will come up, and then the whole thing will be excreted, and it will again form a intact surface like this. Okay. It will come up and form an intact surface like this. I hope this is clear, the process. In amoeba, carbon dioxide and animal and ammonia are the main waste material. These are excreted by diffusion through the general body surface. Okay, so carbon dioxide and ammonia, these are the two main excretory products for amoeba. Okay, contractile vacuole also helps in excretion. Contractile vacuole definitely helps in excretion. The waste present in cytoplasm enters the contractile vacuole, which moves towards the cell membrane. The waste is then thrown out into the water body. Okay. Now, my dear, I hope this is very clear. Nothing is very confusing here. These things are really easy to understand. Okay, they are basic cells doing some basic things. Reproduction in amoeba, there are two types of reproduction which amoeba practices. This one is binary fission and one is multiple fission. First, we will see binary fission. One parent cell is there in which two nuclei, the nuclei will be divided and two nuclei will be formed, and then the whole cell will start dividing with one nuclei on each side, and then the whole cell will be divided in two equal daughter cells. Okay, two daughter cells. Okay, so please excuse me, I'll something problem is there, I'll just back in. Okay, leave, it is good now. Okay, so my dear students, this image is really exciting. As you can see how it will divide in two equal parts. Two nuclei are formed, both the nuclei went to both the sides, and then the whole cell divided into two daughter cells. Isn't it interesting? You see, as you can see, it is trying to divide, and yes, with two different distinct nuclei, it gets divided and forms two daughter cells. Okay. Okay. Amoeba reproduce asexually by a binary fission. Amoeba reproduce asexually by binary fission, not a binary fission, binary fission. The nucleus of amoeba elongates and finally divides into two daughter nuclei. The cytoplasm of the cell then divides, resulting in the formation of two daughter cells. Simple. The nucleus divides and then the cytoplasm divides and then two daughter cells are formed. Okay. Multiple fission is also there. This is what multiple fission looks like. One daughter cell is there, a, which is movable, and a strong cyst is formed, a strong covering is formed on the cell, and then the nucleus divided into many nucleus, and then many small cells are formed, and then all of them are released. We will study this in detail during unfavorable condition. This is important, my dear students. In favorable conditions, amoeba simply divides through binary fission. But when the condition is unfavorable, then they do something different. What do they do? During unfavorable condition, amoeba reproduces by multiple fission. It secretes a thick wall called cyst around itself and becomes inactive. It doesn't do anything, it becomes inactive. Simply a cyst like form structure is formed, strong structure, and it becomes inactive because the condition is unfavorable. The nucleus divides repeatedly and forms several nuclei. Inside that cyst, nucleus divides repeatedly, means multiple times, and forms several nuclei. Okay. Each nucleus then becomes surrounded by a small amount of cytoplasm, because cytoplasm is very essential for any cell. Nucleus cannot survive alone. So each nucleus is then surrounded by small amount of cytoplasm on return of favorable condition. Once the favorable condition returns, the cyst breaks, liberating the daughter cells, each of which grows into a new amoeba. See, when the condition is favorable, cells are normally dividing in binary fission here and there, but when the conditions are unfavorable, it could last for several weeks or days or even months. Okay, so at that time, it forms a cyst and divides internally. And once the favorable condition comes, then this cyst ruptures and all of the cells releases. Okay. Okay, so this was it. This was it, my dear students. I hope you understand. This was not tough. Amoeba is very easy to understand. Simply, and in Protista, we have two more different types of organisms, which are Paramecium and Euglena. Amoeba's functions we understood. It basically works through pseudopodia. Okay, locomotion and ingestion of food, and digestion is done with the help of food vacuoles, and excretion is also done with the help of those vacuoles. Mitochondria is present. Other cell organelles are also present in amoeba. Revise it once more if you want. It will be better if you see this video 2 times. Then most of the things will be will get cleared by only seeing the video itself. Okay. Thank you very much. In our next class, we will study about the kingdom fungi. God bless you all.